Bill, thank you. Hey, thanks Bill and Beth. It's really wonderful to be here. I want to I'm going to present some of what we've been seeing related to oyster mortality in North Carolina, but also push a plug. Bill is leading a session on these mortality issues that a lot of us are seeing across the Atlantic and Gulf coasts from a broader and much more diverse perspective than just my single North Carolina perspective. So stay tuned, but let's talk about what's happening in North Carolina a little bit right now. And before I go that, hi, I'm Tal Ben-Hareen. I'm an assistant professor at the College of Vet Medicine in North Carolina State University. And this work represents a collaboration. A lot of my colleagues, should I talk a little closer to that? Rachel Noble at UNC Chapel Hill, Amy Wilbur at UNC Wilmington, Grant Murray at Duke Marine Lab, and then Jess Small and Corinne Odemard at Virginia Institute of Marine Sciences. Y'all can hear me okay, right? Okay. And just to start, we had a few of these conversations yesterday, but let me talk about the North Carolina industry a little bit. We've seen phenomenal growth in the past decade, especially the past few years. These data are from Eric Herbst, so I haven't seen yet, but you'll see them a little bit. It looks like we need to update them for 2023, but in the past 5 years, we've seen the industry expand quite a bit. 2022, we're up to about $5 million in farm gate value. And with that growth, we're starting to see a lot of challenges, just like any industry growing. Those data, it's hard to get data from growers on losses, but one of the things, we're in really great communication with our USDA, our insurance team, and in 2022, we had estimated about $5 million in insurance claims to match that $5 million in landings. And so with that growth, we're seeing a lot of mortality, and obviously the things that come with it. And this has made some press. We seem to be a hot spot, probably due to the region, which I'm going to talk about in a little bit, but also probably due to the fact that the industry is growing. Me as a pathologist, because we have such consistent mortality across the state, it's been a really nice place for me to build a research program as a pathologist, because it's let me consistently and very reliably sample from oyster populations and sites that I know are going to go through a mortality event. And in doing that, one thing that we do see is really consistent pathology. One thing that we see in our mortality in North Carolina seems to be consistently associated with pathology in the gut, and the diverticula in particular. What you're looking at here are some histology slides. The slide on the left is what look like pretty healthy tubules aside from the massive amount of inflammation that's happening. All those little dark spots are oyster hemocytes. Those are the main immune cells. And then what you see on the right is kind of what happens after that inflammation. Those digestive tubules have sloughed away the cells that are responsible for all the intracellular nutrient assimilation. And it's kind of normal. It happens on a tide. Food comes in, oysters feed, all the other stuff that's associated with the food that isn't necessarily nutritious. Those cells will slough away and get shed out as feces, only to be regenerated on the order of 8 to 12 hours, like our own intestinal cells. What we see associated with these events is for 3-4 weeks, just no cellular regeneration. And we can look at this in a time series. Again, I mentioned we get these sites that have really consistent pathology, mortality year after year, which has allowed me to go out and sample and set up sentinel field sites where I can go out ahead of time and through these actual mortality events. And this is just a summary of just grading of the pathology that we see, both in the diverticula, but also the intestines. This was a mortality event, a site that saw a mortality event in late August. And you can see starting in early to mid-July, digestive diverticular pathology starts to creep up, and it stays really high. In this event in particular, the mortality was first seen on July 21st, in that July 21st sample. So it's like 3 to 4 weeks of pathology before we see mortality. We do see some intestinal pathology associated with it, but it seems to follow the mortality. And I don't show any slides here, but it does seem to be more associated with regular Perkinsus marinus infection. The thing that these animals present clinically, a lot of you have probably seen this, just that whole visceral mass. You can see that, you can see the kind of the back part of that oyster there, the meaty bit of the oyster, that's like the rich in connective tissue and glycogen, kind of implodes inside the shell there. It's really watery. That's kind of the main clinical feature that we see. And then this other histology slide that I'm showing here, this is just a slightly different stain that makes the bacteria pop a little bit. And I do have some arrows on there. Those arrows are pointing to little pink spots, which are bacterial infections. And that's just, I'm not classifying those bacteria, I'm just saying there are bacteria associated with this oyster, there are bacteria associated with even a healthy oysters. But with that pathology, we see a bit of bacteria. And we've done some sequence work starting to characterize what these bacteria are, some of the microbial species that are associated with both healthy oysters and oysters going through these mortality events. And what we're looking at here are some bacterial taxa associated. On the left is sampled from oysters before a mortality event. The middle is during a mortality event. And on the right is after a mortality event. And we definitely get these flips. Before a mortality event, we're more likely to see oysters characterized by taxa like Sphigomonas or Hymenobacter. And we do see this shift towards more Ralstonia, Pseudomonas. I don't think these bacteria are contributing to mortality, or they're not the primary cause of mortality. They seem to be more associated, responding to the pathology itself. A lot of these taxa are like the things that you see in immune-compromised hospital patients, like their hospital bacteria, Ralstonia, Pseudomonas. So I think these are more opportunist secondary pathogens. And so I want to talk about some of the work that we've done. This was some work funded by NCDEQ and North Carolina Marine Fisheries, and this was a collaboration with VIMS and UNC Chapel Hill, UNC Wilmington that I mentioned. And there's this open question about the relative contribution of the underlying genetics, the broodstock origin, or triploidy. And so we've made some paired crosses where we've taken some of the common lines in production in Virginia that come out of Virginia, like Henrys, and we cross them with both diploid Henrys, but then we also cross them with GNL tetraploids. And we have a small breeding program at UNC Wilmington where we're making North Carolina-derived oyster lines. So we took those same, we did the same crosses, where we crossed a Hewlett's Creek, a North Carolina-derived diploid with the Henry diploid. And we call that a North Carolina diploid. It's half Henry, so it's not really North Carolina. But then we also crossed those North Carolina diploids with the GNLs and made what we call a North Carolina triploid. And we deployed it, can I go back? Yeah, we deployed this to 8 sites, 6 sites in North Carolina, and then 2 in Virginia. The 2 in Virginia are VIMS, the York River, but also Cherrystone. And I'm gonna talk through some survival, and I just wanna walk through what we're looking at here. These are each of the colored lines represent one of those 4 oyster genetic lineages that I mentioned. We have the Virginia-derived, the Henry diploids and triploids, and the North Carolina-derived diploids and triploids. And for each of these curves, you see 2 lines. And all that means is that we go out, at best we can get out to a site once a week. Sometimes I have some wonderful students that would love to be out in the field every day if they can, and they've pitched some of this daily sampling, but we really can't go out, logistically can't go out and sample daily. And so for each of those 2 lines, what those 2 lines mean is each time we go out in the field, we count the number of oysters that are alive, the number of dead. If we find an oyster dead on some given day, but we counted it alive 2 weeks ago, we know that it died sometime between 2 weeks ago and today. And that's what those 2 lines are showing. It's just that variation and that uncertainty when it actually died, but we just know it died between that interval. And so there's 2 for each. And these are gonna present a little bit small. These are those 8 sites, but I just wanna walk you through some general patterns of what we're seeing. Across those 8 sites, we have 2 sites that suffered what we call a mortality event. And when I mean mortality event, I mean it's an event, near 100% mortality. And so you can see it's which ones are we looking at? This is the top rightmost, Core Sound 3, was one of the sites that we consistently and reliably see mortality year after year. The other site that had what's a mortality event is Bogue Sound Seamast, and I'll take all the credit for that. This is my lease. And this is just a site, it's in a cove, there isn't a lot of flow, and then pretty much every year we see an August mortality event. It's not the best place to grow oysters, but it's really convenient for me because it's just outside my office. We grow great oysters in the winter, but I need to get them out of the water by May. But so in that lease my lease that consistently sees mortality, we had a May mortality event, and then the kind of later mortality event in that Core Sound 3 site. When there's an event, when mortality is really bad, the genetic line or triploidy doesn't matter. It's bad everywhere. But across these other sites, what we see is some patterns start to emerge. And one, those Virginia diploids were the worst performing line across the board. Virginia triploids were the worst performing line across the board. Diploids. The North Carolina diploids were the best performing line. And then the other two, the Virginia triploids and the North Carolina triploids, were somewhere intermediate. And what that's telling us is that it's not necessarily ploidy that's driving the mortality here. It looks to be the geographic or genetic origin of those lines seems to be the more important driver. We know that there's a lot of variation across sites. We have these sites that pretty consistently see pathology. And we have sites that pretty consistently don't see any pathology and no mortality. The sites that do see mortality seem to be lower in the estuary, high salinity sites. And what we're starting to do is move some of our farms and our own research sites up the estuary. And we did a manipulative experiment last summer where we started a cohort of oysters at a really high risk, high mortality site, and then moved it, one up the estuary. I don't want to reveal the names of the sites here to keep that to anonymize the growers that we work with, but I'll call it a promising up estuary site. And then it isn't just salinity. We have some sites that are really high salinity, high flow, any number of factors that seem to do just fine. So while we also moved that cohort up estuary, we also moved it to the magic lease, high salinity. And this is what we're looking at here. The site that reliably sees mortality died like clockwork on Mother's Day. But then we moved it up estuary, did fine. Moved it to the magic site, did fine. So it seems like, taking all this, it seems like summer mortalities, I'm not saying anything new here, but it seems to be a G by E, a genetics by environment interaction. I'm at a college of vet medicine. I talk about this and many other diet, disease and pathology across various tasks with my veterinary colleagues. And in veterinarians also, they always complain about not being able to speak to their patients. If you think about a dog, at least you can talk to the owner, or you can palpate a lymph node. We're talking about oyster rocks here. It's really hard to talk to an oyster, or you can talk to a farmer. But thinking about, and not just that, in veterinary medicine, there's decades of case reports across various things that may go wrong. And we just don't have that background to draw from. But if we were to if we were to do rounds on this and talk through differential diagnosis, we can kind of classify these as two things, and two really broad general things. We're not even close to scratching the surface here. But based on what we're seeing, the inflammation, the persistent digestive diverticulate pathology, that G by E interaction, we can kind of classify this as an autoimmune, autoinflammatory type disease, something like, the analog would be like Crohn's disease in humans. And then there's also, can't rule it out, the various microbial infections, and that's supported by some of the molecular data that we have. We're not there yet. And like I said, the microbial data that we have right now is more indicative of like a secondary process responding to something else going on. So our ongoing work where we're still classifying this pathology, how it happens across different sites, across all different genetic lines, and what we're really moving towards is how that varies across those different genetic lines and finding genetic lines that seem to work a little bit better. And then the other thing, I did mention that we have some promise about moving up estuary, and there's other stuff we can do, like the timing of deployment, timing of harvest, keeping those larger oysters out of the water, especially at high salinity sites during warm summer months. So thanks to various funding agencies, NC Collaboratory, Sea Grant, NCDEQ. There's my contact information there. I'm going to be here all day for anybody who wants to chat, and I'm happy to answer any questions.
All right. Thank you, Tal. Do we have any questions for Tal?
Yeah, Leslie.
What about gill pathology?
Can you repeat it?
Yeah, Leslie asked a question about gill pathology. And the reason why Leslie asked is Ryan Carnegie's here, and the sampling that Ryan's done, Virginia and elsewhere, not always, but some inflammation, hemocytosis in the gills, erosion of gill epithelia is commonly associated with this. We don't see it to the same extent that we see diverticulate pathology. I do see some inflammation in the gills, and we say that's more like a generalized kind of multifocal immune response, but we don't see the same gill pathology that's been seen elsewhere.
All right. Other questions for Tal?
Did you have a chance to look at the surrounding water microbiome composition?
In the water?
Yeah.
Yeah.
Well, potentially their food.
Oh, yeah. Yeah, we don't see a lot, the food, we don't see a lot of microbial changes in the food. What we see timed with, obviously Vibrios always get implicated in these kinds of events. What we see, not during the mortality, but during the time when we see a lot of that diverticulous sloughing, a lot of seawater Vibrios. The seawater Vibrios seem to be responding to what's in the oysters as opposed to the other way around.
All right. So,
Thanks, everyone.
folks, we have, thank you, Tal. Let's give him another round of applause.